Have you ever wondered how your body fights off an infection like instantly? I mean, long before your main immune cells even get the memo that there's a problem? Well, today we're diving into that exact system, the incredibly fast, powerful, and honestly kind of brilliant complement cascade. So, how does it do it? How does it have this lightning fast reaction time? The secret isn't about creating a new response from scratch. It's about activating a system that's already there, circulating in your blood, just waiting for a trigger. And that's the complement cascade. The best way to think about it is like a silent alarm system for your entire body. It's not just one thing, it's a whole team of proteins all working together to set off a chain reaction the second an intruder is detected. And what's really wild is that the complement system isn't made of cells at all. Nope, it's a group of about 30 different proteins that are just floating around in your blood, completely inactive. You can think of them like tiny sleeping soldiers, just waiting for the signal to wake up and get to work. Okay, so once that signal comes, what do? Well, they have three main jobs. First is something called opsonization, which is basically just a fancy word for painting a giant eat me sign on an invader so other immune cells can easily find it and, eat it. Second, they recruit backup. They release these chemical signals that are like a flare gun, calling in reinforcements. And third, they go for the direct kill with cell lysis, building a weapon that literally punches holes in the bad guy. Game over. So, how does this alarm get tripped in the first place? It turns out there are three main ways, or pathways, to kick this whole thing off. You've got the classical pathway, which needs antibodies to point out the target. Then there's the alternative pathway, which gets triggered directly by the surface of a pathogen itself. And finally, the lectin pathway, which is super cool. It recognizes specific sugar patterns that are only found on microbes. So three totally different tripwires, but they all lead to the same massive immune response. All right, let's start with the classical pathway. Think of this one as the precision, targeted approach. It relies on work that your more specialized immune system has already done. So here's how it goes down. First, your antibodies, specifically types IgG or IgM, find their target on an intruder's surface and latch on. The moment that happens, a complement protein called a C1q sees that antibody and grabs onto its base. That little handshake is the signal that kicks off the entire cascade. And just like that, the alarm is officially on. So as the alarm is blaring, the cascade starts churning out these really crucial molecules. C3b is a master opsonin. It's that paint we were talking about. It completely coats the target, making it look absolutely delicious to your cleanup cells. At the same time, you get molecules like C3a and C5a. These are the flare guns, the chemical signals that shout for help, and bring a whole swarm of other immune cells rushing to the scene. But hang on, what if you've never encountered a pathogen before? You wouldn't have any antibodies ready to go, right? Well, that's where the other pathways come in. Let's look at the alternative pathway. This is your rapid response unit, a much older and more immediate line of defense. The amazing thing about this pathway is that it doesn't need antibodies at all. It can be triggered directly by common patterns found on pathogens, like something called lipopolysaccharide, or LPS, on bacterial walls. This makes it incredibly versatile and a fantastic first responder. It can even be set off by stuff like cobra venom. This pathway is basically a biological shortcut, and it's not the only one. The lectin pathway, which we mentioned earlier, works in a similar way. Instead of recognizing general danger signals, it has these special proteins that are like little sugar detectives, and they're hunting for specific carbohydrate patterns you only find on microbes. Both of these are just brilliant ways to sound the alarm without waiting for antibodies to be made first. So let's just put it side by side. The classical pathway is methodical, specific, it uses antibodies as its guide. Then you have the rapid responders, like the alternative and lectin pathways. They are all about speed, jumping past those first few steps to get a defense going immediately. It's like having both a highly trained sniper and a frontline guard who's ready to react to anything. Now this is where it all comes together. It doesn't matter how the cascade starts, whether it's the classical, alternative, or the lectin pathway. They all merge and head towards one single final goal, building the ultimate weapon. And that weapon is called the Membrane Attack Complex, or MAC for short. And let me tell you, it is exactly as intense as it sounds. These complement proteins literally self-assemble into a structure that acts like a molecular drill, and its only job is to bore a hole right into the enemy cell's membrane. For the intruder, the result is absolutely catastrophic. The MAC forms a stable pore, a tunnel, right through the cell's protective barrier. Water and salts from the outside rush in uncontrollably. The cell can't handle the pressure, and it swells up and bursts. This is called cell lysis. It is a direct, lethal, and incredibly effective way to take out the trash. Now you're probably thinking, a weapon that powerful needs some serious safety controls, right? You can't just have these molecular drills running amok. And you are absolutely correct. Your body has a ton of regulator proteins to keep the system in check. For example, the C1 esterase inhibitor acts like a safety brake on the whole process. When that brake doesn't work right, like in a condition called hereditary angioedema, the system can fire off randomly, causing all sorts of problems. It's a perfect example of how critical that balance is. And this brings us to the final and maybe most important question. We've seen how this system can utterly demolish invaders with amazing efficiency. So what stops this incredible weapon from turning on our own healthy cells? The answer, it turns out, is in a whole other layer of elegant safeguards on our own cell surfaces that tell the system, hey, I'm one of the good guys. It just goes to show you the beautiful complexity of our immune system, always balancing immense destructive power with the delicate art of self-preservation.